Hey everyone, and welcome to Kognito Key. I am absolutely thrilled to have you join us today. Imagine, if you will, a pristine, refreshing, ice-cold glass of fresh milk. Now imagine a completely curdled, sour, spoiled mess right next to it. What exactly went wrong here? Well, today in this explainer, we are going to investigate a literal microscopic crime scene. You see, fresh milk is like a luxurious five-star, nutrient-rich resort, and unfortunately, it naturally attracts some very uninvited microbial guests. We're going to uncover exactly who these culprits are, how they absolutely ruin our milk, and the brilliant scientific methods we use to catch them in the act. So, let's dive right into the facts. The spoilage of milk is officially defined as any undesirable change or deterioration in its quality. We are talking about an unpleasant appearance, a weird color, a foul odor, or that awful bitter taste you never forget. But why is milk so incredibly susceptible to this kind of attack? Well, milk has a very high water activity and an incredibly complex biochemical makeup. It's packed with proteins, fats, and carbohydrates. For us, that means nutrition. But for microorganisms, it makes it the absolute perfect culture medium to grow and multiply. It provides the ultimate scene of the crime for microbial growth. Now, what's really fascinating is how we can actually break down the conditions of our crime scene into two totally distinct categories: intrinsic and extrinsic factors. Think of intrinsic factors as the internal biological motives of the milk itself. This is its moisture content, its pH levels, its nutrients, and even its natural antimicrobial constituents. Extrinsic factors, on the other hand, are the external environmental opportunities that actually allow the spoilage to happen. This includes the temperature the milk is stored at, the relative humidity, the availability of oxygen, and how different microbes interact with each other. Simply put, without the right external environmental opportunities, those internal motives just don't lead to a crime. All right, let's bring in our first lineup of suspects and look at the actual biochemical crimes they commit. First up, we have souring, which is most often caused by bacteria like lactobacillus. They give the milk that sharp, bitter taste and cause it to form curds. Next, there is sweets curdling, brought to us by microbes like bacillus. These sneaky guys actually shift the milk's pH to alkaline, which causes the proteins to coagulate. But honestly, the most dramatic crime of all is gas production, typically caused by coliforms or Clostridium. This causes a phenomenon known as stormy fermentation. It literally results in an explosion of curds right at the top of the milk. Talk about an aggressive microbial takeover. Beyond just souring and gas, we also have some highly visual and, frankly, pretty gross crimes of spoilage. Certain bacteria, like alkylogenes, cause something called ropiness. They produce this slimy capsular material that turns the milk into highly viscous, sticky, silk-like threads. It is exactly as unappetizing as it sounds. Then we have striking color changes. Depending on the specific bacteria, like various Pseudomonas species, your milk could turn bright blue, yellow, red, brown, or even a weird grayish color. Finally, we have dairy molds like penicillium. These don't just give the milk a fuzzy, moldy appearance, they can actually produce really dangerous toxins. So, an absolutely crucial point to understand here is that not all of these bacteria strike under the same conditions. They actually have vastly different temperature preferences. On the colder side of things, we have psychrophilic bacteria. These are the cold-loving culprits that thrive below 10 degrees Celsius. They often degrade proteins and fats to cause terrible off flavors, even when your milk is sitting right there in the fridge. On the completely opposite end, we have the thermophilic, or heat-loving bacteria, which flourish in hot environments between 55 and 65 degrees Celsius. Knowing their preferred operating temperatures is exactly how we begin to build our thermal defenses against them. Of course, we aren't just worried about strange colors and sticky threads. Some of these suspects are far more dangerous to us. We're talking about pathogenic types, microbes that can cause serious illness, potentially transmitting severe diseases like tuberculosis through Mycobacterium tuberculosis, or even anthrax via Bacillus anthracis in raw milk. Which brings us to a really vital question. How do we actually detect these microbial culprits and dangerous pathogens before they ever reach our breakfast tables? It's time to transition to the scientific interrogation phase. Let's open up the Dairy Detective's basic toolkit. The standard plate count, which is also known as the viable count method, is a highly effective, direct way to visually assess live bacteria. Here's how we do it. First, we take just 1 milliliter of the milk sample and dilute it. Then, we spread half a milliliter of that sample over solidified agar media. We pop those plates into an incubator for 24 to 48 hours at a nice, warm 35 to 37 degrees Celsius. Finally, we count the bacterial colonies that grow. The rule in the lab is crystal clear. If we count more than 300 colonies, that milk is officially deemed unpotable, meaning there is absolutely no way it is safe to drink. Now, if we need answers a bit faster, we can use the Methylene Blue Reductase test, which is actually incredibly clever. We add a distinct blue dye straight into the milk. As the bacteria breathe and consume the oxygen, they reduce the dye, which causes that blue color to literally disappear. It's almost like a quick confession from the microbes. The faster the color drops, the more bacteria are in there working. If that color vanishes in under 2 hours, we know the milk is of very poor quality. But if the milk holds on to its blue color for over 8 hours, it tells us the bacterial load is incredibly low and we are looking at the absolute best quality milk. Finally, we absolutely must verify that the dairy industry's thermal defense systems, like pasteurization and sterilization, actually did their jobs. We do this through 2 really cool key tests. First up is the phosphatase test, which checks for proper pasteurization. Phosphatase is an enzyme that's naturally present in milk, but it gets destroyed by proper heat levels. So if our test turns blue, it means the enzyme is deactivated and the milk was perfectly pasteurized. Second, we have the turbidity test, which checks for sterilization. If we boil a treated sample and turbidity, which just means cloudiness, appears, it means coagulable proteins are somehow still present, and the milk was not sterilized properly. It's a brilliant set of chemical checkpoints. And that brings us to the end of our microbial investigation. Understanding these biochemical crimes and properly executing these lab tests is absolutely vital for ensuring the safety of the dairy products you and I consume every single day. But, it really makes you wonder, if milk is such a perfect environment for these microscopic invaders, what other foods sitting in your fridge right now are currently hosting an invisible microbial war? I hope you found this explainer as fascinating as I did. If you enjoyed this deep dive into the microscopic world, please make sure to subscribe to the channel and like and share this video so we can keep bringing you more incredible insights here at Kognito Key. Until next time, stay curious and keep learning.